We are talking about gonorrhoeae, cultivation of gonorrhoeae. They don't grow necessarily, including gonorrhoeae, don't grow on ordinary media. They need media with the proteins, and they grow better at the presence of CO2, 3 or 10%. They develop S-type colonies without color. Biochemical properties. They have no biochemically active. They are able to ferment just one sugar, glucose, and no proteolytic properties. Antigens. Gonorrhoeae contain capsular and somatic antigens. Pili protein. Pili for adhesion, they have got cell wall proteins and cell wall glycolipids. They are very complicated antigenic structure. They have a lot of serogroups and serovariants. Pathogenicity factors. Number one, pili. In the absence of the strain becomes subvalent. Pili or fimbria for adhesion, outer membrane proteins, endotoxin because of their gram-negative bacteria. They have complex of protein and lipooligosaccharide in the cell wall. And then they produce enzymes, proteases, hyaluronidase, and bacteriocides. And they are optional intracellular parasites. Facultative intracellular parasites, actually, they are able to survive inside the cell. So difficult to treat such patients because of gonorrhoeae are able to survive inside the cell. And chronic gonorrhea is developed very often. Epidemiology, the source of infection is a sick person, transmission ways contact, sexual contact, so contact at household, and again, this gonorrhoeae are transmitted from mother to newborn. They don't go through placenta, but the newborn is able to be infected due to contact with infected genital tract of mother. And gonorrhoeae have affinity to mucosal membrane of genital tract. They cause gonorrhea, and they are able to multiply on the mucosa of eye. On eye, they are able to cause conjunctivitis. Conjunctivated names the venereal of newborns and an adult's conjunctivitis with pus. So they attach by using of fimbria to the epithelial cell receptors, then penetration inside the cell by endocytosis, then incomplete phagocytosis, and then release of endotoxin because the temperature. And they are able to infect other organs at the same time. Clinical forms. So they out damage to the genitourinary system, urethritis, prostatitis in men, cervicitis, adnexite in women, and extragenital diseases, lesions of the rectum and pharynx, proctitis, pharyngitis, and venereal of newborns, and conjunctivitis of adults. The disease can become chronic, leading to infertility. Chronic inflammation of reproductive organs leads to infertility sometimes. Non-treated gonorrhea and untreated venereal leads to blindness. Immunity is not formed because, again, gonorrhoeae have very complicated antigenic structure, and the character of immunity is not species specific. Reinfection causes the secondary infection by new threat leads to a infection. Microbiological diagnosis. A material for research in men, urethral discharge, in women discharge of the urethra, cervix, and rectum. And in case of conjunctivitis, pass from the eyes. Microbiological diagnostic method. Bacterioscopic, microscopical method, bacteriological method, cultivation, serological, detection of antibodies in case of chronic gonorrhea, and molecular biological method. PCR is used for detection of nucleic acid. Treatment. Really, strains of gonorrhoeae are resistant to many antibiotics, and it is better to isolate pure culture and to do any antibiotic sensitivity test. And every patient should be treated personally in accordance with results of antibiotic sensitivity test. There are just, for example, cephalosporins, macrolides, because they go inside the cells, like erythromycin, clarithromycin, azithromycin, fluoroquinolones, and so antibiotic sensitivity test, it is required for each patient. For treatment of chronic gonorrhea, inactivated vaccine is used. I told you that for prevention of gonorrhea, there is no vaccine for prophylaxis. No vaccine all over the world, but vaccine is developed. It is inactivated vaccine, and it is used for chronic gonorrhea. The aim is to stimulate immune system, and it works. So very rarely it is an exception, because of vaccines mainly are used in 99% are used for prevention, for specific prophylaxis. Absolutely healthy people should be vaccinated for any case, but there are a few diseases, if they are chronic, inactivated vaccines are used for treatment. Staphylococcal vaccine, gonococcal vaccine, and then we will study brucellosis for treatment of chronic brucellosis. Inactivated vaccine is used, and herpes viral infection. Inactivated vaccine is used for treatment of herpes viruses. Okay, not for elimination, but anyway, it helps. Prophylaxis, the same rules as for all other sexually transmitted disease, the use of condoms, the exclusion of accidental sexual intercourse, control over the recovery of patients. That's all. Have you any questions?
Yes, please, concerning the sensitivity test against gonorrhea. Why? Yeah, sensitivity test, antibiotic sensitivity test. If it's bacteria, why should we do for each individual?
Because of listen, because of every patient is infected by certain strain of the Neisseria gonorrhoeae. You know, we are talking about species in general, but in real life, we are talking about strains. Those are circulated between people, and strains, in spite of they belong to one species, Neisseria gonorrhoeae, strains are differ by sensitivity to antibiotics. So from every patient, PU culture should be isolated, and we should use any antibiotic sensitivity test. This diffusion test, dilution test, Alamar test. Now there are many e-tests. Many tests are used to know exactly what antibiotics these bacteria are sensitive to. For one patient, for another patient, PU culture should be isolated. And again, we have to check what antibiotic this bacteria, this strain of Neisseria gonorrhoeae is sensitive to. The problem is that strains, in spite of they belong to one species, they differ by sensitivity to antibiotics. Do you understand?
Thank you. Yes.
In case of Treponema pallidum, did you notice I told you we are lucky? Fortunately, Treponema pallidum did not develop resistance to penicillins. And it is possible penicillin was the first antibiotic. It was developed at 1940s, and it was used almost 80 years. And many bacteria did develop resistance to penicillin, except Treponemas. But in case of Treponema pallidum, we are not going to do antibiotic sensitivity test, even the PU culture is not isolated. But sometimes it is necessarily. In case of gonorrhoeae, it is necessarily. Do you understand? Any questions?
Yeah.
Have you any questions? If no, I will finish the lecture, and next lecture will be in 2 weeks from now. No, next Friday. Next from next, okay?
Yes,
Yes,
Doctor,
yes.
please. Can I find these materials in the portal or not?
It is the lecture is thank you so much. The lecture is placed on educational portal, and now all students should go to the educational portal and answer tests for lecture. And I didn't check you now. I will check attendance of the lecture in accordance with answers on educational portal. If student did answer test for lecture, he did attend the lecture. If not, I will write absent. Okay, leaders, you are responsible. Could you inform all group mates, please? During this day, they should answer tests for the lecture, okay?
Until 12 o'clock. Yeah?
By
Until-
groups, it is written, groups are written, so don't mix, please. Answer in accordance with your group number, and I will check attendance. I will check answers for test, okay?
Okay, thank you. Have a nice day.
Leaders, you are responsible. Bye-bye.
Bye-bye, doctor.
If you have no questions, thank you for attention and goodbye. Goodbye.
Goodbye, doctor.
Goodbye. Bye-bye. And fourth group, we will meet tomorrow for laboratory class.